Friends, my name is Mr. Desle SD, working Mahatma Gandhi Vidya Mandirs, Janta Vidyalay and Junior College, Manur, Taluka Kalawan, District Nasik. My subject is biology and we are going to study now chapter number 4, molecular basis of inheritance. The last slide or video, we are going to study now characteristics of genetic code. We teach 10 characteristics of genetic code. Now, today we are going to study now next topic, that is mutation and genetic code. Mutation is the phenomenon in which sudden change in the DNA sequence takes place. It results in the change of genotype. Along with recombination, mutation is raw material for evolution, as it also results in variations. During mutation, possibility of loss or gain of a segment of DNA result in alternation in the chromosomes. Mutation can also occur due to change in a single base pair of DNA. This is known as point mutation. Example, sickle cell anemia. Sickle cell anemia. Deletion of base pair of DNA causes frame shift mutation or deletion. Mutation inserted or deletion of one or two base changes the reading frame from the point of insertion or deletion. Insertion or deletion of three or multiple of three bases result in insertion or deletion of amino acids, and reading frame remain unaltered from that point onwards. Transfer RNA, the adapter molecules. The scientists consider that there has to be a mechanism in which transfer RNA will read the codon and also simultaneously binds with the amino acids. As amino acids does not have any special capacity to read the codon, so transfer RNA considered as an adapter molecules. These roles of transfer RNA was understood much later. In the diagram of transfer RNA and cloverleaf structure of transfer RNA possess an anticodon loop that has bases complementary to the codon. It is called anticodon. It also amino acids, acceptor and having unpaired CCA bases to which amino acid binds. For every amino acids, there is specific transfer RNA. Initiator transfer RNA is specific for methionine. There are no transfer RNAs for stop codon. In the actual structure, the transfer RNA molecule looks like inverted L, that is three-dimensional structure. The next subtopic, that is translation, that is protein synthesis. Translation is the mechanism in which codon of messenger RNA are translated and specific amino acids in a sequence from a polypeptide on ribosomes. All types of proteins are synthesized by the cell within itself. Process of translation requires amino acid, messenger RNA, transfer RNA, ribosomes, ATP, magnesium, iron, enzymes, elongation, translocation, and release factors. Number one, amino acid from raw material for protein synthesis. About 20 different types of amino acid are known to form proteins. These are available in the cytoplasm. Number second, DNA controls synthesis of proteins having amino acid in specific sequence. This control is possible through transcription of mRNA. Genetic code is specific for particular amino acids. Number 3. RNA serve as intermediate molecules between DNA and proteins. Number 4. Ribosomes serve as site for protein synthesis. Each ribosome consists of large and small subunits. These subunits occur separately in cytoplasm. Only during protein synthesis, these two subunits get associated together due to the magnesium ion. A ribosome has one binding site of messenger RNA and third binding site for transfer RNA. They are P sites, A sites, E sites. Only first transfer RNA amino acid complex directly enter P sites of ribosomes. Next topic, that is mechanism of translation. It involves three steps. These three steps are: number one, initiation of polypeptide chain. It consists of three substeps. That is, first one is A. Activation of amino acid is essential before translation initiates, for which ATP is essential. Small units of ribosome binds to the messenger RNA at fifth end. Initiator codon AUG is present on messenger RNA, which initiates the process of protein synthesis. Initiator transfer RNA binds with initiation codon by its anticodon through hydrogen bonds. It carries activated amino acids, methionine or hormel methionine. Next, B. Now the large subunit of ribosomes join with the smaller subunit that's require magnesium ion. Next, C. Initiated charge transfer RNA occupies the P sites of ribosome and A sites is vacant. Number second, that is elongation of polypeptide chain. During this process, activated amino acids are added one by one to first amino acids. Amino acid is activated by utilizing energy from ATP molecules. These amino acids bind with amino acid binding sites of transfer RNA. This result in formation of transfer RNA amino acid complex. Addition of amino acid occurs in three steps. That is, number one, codon recognition. Amino acetyl transfer RNA molecules enter the ribosome at A sites. Anticodon binds with the codon by hydrogen bond. B. Amino acid on the first initiator transfer RNA at P sites and amino acids on T RNA at A sites joined by peptide bond. Here enzyme ribosomes act as a catalyst. At this time, first transfer RNA at P sites is kicked off. Next, C. Translocation. The transfer RNA at A sites carrying a dipeptide at A sites moves to the P site. This process is called as translocation. In translocation, both the subunits, the ribosomes, moves along the relation to transfer RNA and mRNA. Hence, transfer RNA carrying dipeptide now gets positioned at P sites of ribosomes, making A sites vacant. At this site, then next charge transfer RNA molecules carrying amino acids will be received. During these processes, first uncharged transfer RNA is discharged from E sites. This process is repeated as amino acid are added to polypeptide. It takes less than 0.1 second for formation of peptide bonds. Third charge transfer RNA with its amino acids arrives at a sites of ribosomes. Anticodon and codon bind by hydrogen bond. Polypeptide bond is formed. Second transfer RNA is discharged from P sites to E sites and leaves of ribosomes. So, the event like arrival of transfer RNA amino acid complex, formation of peptide bond, ribosomal translocation, and removal of previous transfer RNA are repeated as ribosome moves over the messenger RNA. All the codons of on messenger RNA are exposed one by one for translocation. And this translocation of protein synthesis consists of initiation, elongation, and termination as shown in the figure. Substage number 3, that is termination and release of polypeptide. At the end of messenger RNA, there is a stop codon. It is exposed at the A site. It is not read and joined by anticodon of any transfer RNA. The release factor bind to the stop codon, thereby terminating the translation process. The polypeptide is now released in the cytoplasm. Two subunits of ribosomes dissociate. The last transfer RNA is set free in the cytoplasm. Messenger RNA also has some additional sequences that are not translated and are referred as untranslated region. The untranslated are present at both fifth end and third end. They are required for efficient translation process. Finally, messenger RNA is also released in the cytoplasm. It gets denatured by nucleus immediately, hence messenger RNA is short-lived. Next topic, that is regulation of gene expression. It is the multi-step process by which a gene is regulated and its product is synthesized. Thus, gene expression result in the formation of polypeptide. Gene expression process is regulated at different levels. In eukaryotes, the regulation can be at different levels. Number one, transcription level. Number second, processing level. Number three, transport of messenger RNA from nucleus to the cytoplasm. And number four, translation level. Gene of a cell are expressed to perform different functions. For example, an enzyme beta galactoside is synthesized by E. coli. It is used for hydrolysis of lactose into galactose and glucose. In E. coli, bacteria do not have lactose in the surrounding medium as a source of energy. Then enzyme beta galactoside is not synthesized. So it is the metabolic or physiological or environmental condition that regulate expression of genes. The development and differentiation of embryo into an adult organization is also a result of coordinated regulation or expression of several sets of genes. Now, one has to understand that know the mechanism by which the organism regulate gene expression in response to changes in the environment. If so, whether single mechanism exists for regulation of the expression of different genes, sets of genes, or different genes are regulated by different mechanism. Certain bacteria like E. coli adapt to their chemical environment by synthesis contain enzymes depending upon the substrate present. Such adaptive enzyme is called as inducible enzyme. A set of genes will be switched on when there is necessity to metabolize a new substrate. This phenomenon is called as induction, and small molecules responsible for this, known as induced, it is positively controlled. Next topic, that is operon concept. Operon concept is transcriptional control mechanism of genetic regulation. Francisco Jacob and Jacques Monod in 1961 explained that metabolic pathways are regulated as a unit. For example, in E. coli, when lactose sugar is provided to the culture medium, such induced production of three enzymes necessary for digestion of lactose. The enzymes are beta galactosidase, digest lactose into galactose and glucose. Second, beta galactoside permeates, that is, permits lactose molecules to enter into the cell. Number three, transacetase, that is, transfer an acetyl group from acetyl CoA coenzyme to galactoside. Synthesis of these three enzymes is controlled by a long segment of DNA known as operon. It consists of an operator sites O and three structural genes Z, Y, and A. The action of structural gene is regulated by operator sites with the help of a repressor proteins. Repressor proteins is produced by the action of genes known as regulator genes. The gene expressed depend on whether operator is switched on or switched off. If the operator is switched on, the three genes Z, Y, and A are transcribed by RNase polymerase into a single mRNA. Each structural gene is generally known as cistrons, and the transcribed long messenger RNA offering various cistrons is known as polycistronic. Switching on or switching off of the operator is achieved by a protein called as repressor. When this protein is attached to the operator and blocks it, the switch is tuned off and structural genes are not expressed. Next topic is lac operon. Lactose or lac operon of E. coli is inducible operon. The operon is switched on when a chemical inducer lactose is present in the medium. Lac operon consists of following components. Number 1, regulator gene. Number 2, promoter gene. Number 3, operator gene. Number 4, structural gene. Number 5, inducer. It is not a component of operon. Number 1 component, that is regulator gene. This gene control the operator gene in cooperation with an inducer present in the cytoplasm. Regulator gene precede the promoter gene. It may not be present immediately adjacent to operator gene. Regulator gene produces a protein called repressor protein. Repressor binds with operator gene and repressor its action. It is called as regulator proteins. Number second, promoter genes. This gene precedes the operator gene. It is present adjacent to operator gene. The promoter genes marks the site at which the RNA polymerase enzyme binds. When the operator gene is turned on, the enzyme moves over the operator gene and transcription, the structural genes start. Promoter gene's base sequence determines which strand of DNA acts as template. Number 3, operator genes. It precedes the structural genes. This controls the functioning of structural gene. It lies adjacent to the structural genes when operator gene is turned on by an inducer. The structural genes produce messenger RNA. Operator gene is turned off by a product of refresher repressor genes. Number fourth component, that is structural genes. When lactose is added to the E. coli culture, the structural genes catalyze messenger RNA, which is turn produces polypeptide on the ribosomes. The polypeptide forms act as enzyme to catalyze lactase in the cell. There are three structural genes in the sequence, that is Z, Y, and A. Enzyme produced are B-galactosidase, B-galactoside permease, and trans-acetylase respectively. Next topic is role of lactose. A few molecules of lactose enter into the cell by an enzyme permease. A small amount of these enzymes is present even when apeiron is switched off. A few molecules of lactose act as inducer and bind to repressor. This repressor inducer complex fails to join with the operator genes, which is then turned on. Structural genes produce all enzymes. Thus, lactose acts as an inducer of its own breakdown. When the inducer level falls, the operator is blocked again by repressor. So, structural genes are repressed, inactivated again. This is negatively feedback. Next topic, that is genomics. The term genome is the total genetic constitution of an organism. Alternatively, it is a complete copy of genetic information, one complete set of chromosomes of an organism. The term genomics is the study of genomes through analysis, sequencing, and mapping of genes, along with the study of their functions. The sequencing of yeast, Drosophila, and mouse genome was done in order to facilitate comparative studies between human and other organisms commonly used for genetic studies in laboratory. Several additional genomes are now either activated being sequenced or strongly considered for sequencing. These include several microbes, bees, tomatoes, and other crops. Genomics study may be classified into two types. Number one, structural genomics. It involves mapping, sequencing, and analysis of genome. B. Functional genomics. It deals with the study of functions of all gene sequences and their expressions on organism. Next topic, that is application of genomics. Structural and functional genomics is best for the different purposes in the improvement of croplands, human health and livestock. The knowledge and understanding acquired from genomics research can be applied in the number of different sectors, including medicines, biotechnology, and social sciences. It helps in the treatment of genetic disorders through gene therapy. Number one, genomics is used in agriculture to develop transgenic crops having more desirable characters. Number second, genetic marker developed in genomics have applications in forensic analysis. Number three, genomics can lead to introduce new genes in microbes to produce enzymes, therapeutic proteins, and even biofuels. Next topic is Human Genome Project. The Human Genome Project was initiated in 1990 under the International administration of the Human Genome Organization. The project was coordinated by the US Department of Energy and National Institute of Health. International partner in the United Kingdom, France, Germany, Japan, India, and China. The Human Genome Project formally began in 1990 and was completed in 2003. The Human Genome Project is the multinational research project to determine the genomic structure of humans. The main aims of project are: number one, aim mapping an entire human genome at the level of nucleotide sequences. Number second, aim to store the information collected from the project in databases. Number three, to develop tools and techniques for analysis of the data. Number four, transfer of the related technologies to the private sector such as industries. Five, taking care of the legal, ethical, and social issues which may arise from project. HGP was closely associated with rapid development of new areas in biology called as bioinformatics. The work of Human Genome Project has allowed researchers to begin to understand the blueprints in buildings and constructing the human genome. As researchers learn more about the functions of genes and proteins, this knowledge will have a major impact in the fields like medicine, biotechnology, and the life sciences. Therefore, HGP is very important. HGP was to provide a complete and accurate sequence of 3 billion DNA base pair that makes up the human genome and to find out the estimated numbers of human genes. Now about 33,000 genes have been estimated to the presence in human. The project was also aimed to sequence the genomes of several other organisms such as bacteria, example E. coli, Drosophila, plants, mucus, muscalis, etc. Complete genome sequence of this model organized will be used to for the comparatively studies that will allow researcher to study gene function in these organisms. Next topic is DNA fingerprinting. Genes present on chromosomes are responsible for determining characters of organism as well as for the inheritance of characters. Due to recombination of paternal and maternal gene, we differ from our parents. Difference also arises due to the infrequent mutation that occurs during gamete formation. Due to all these factors, every individual has its unique genetic makeup, which may be called as fingerprint. The technique developed to identify the persons that the help of DNA restriction analysis is known as DNA profiling or DNA fingerprinting. The technique of fingerprinting was first given by British geneticist Dr. Alec Jeffrey in 1984. DNA fingerprinting technique is based on identification of nucleotide sequence present in the wonder molecules. About 99.99% of nucleotide sequence in all persons is same. The length of the region having VNTRS is different in each individuals and hence is the key factors in DNA profiling. Steps involving in DNA fingerprinting are as follow. Number one, isolation of DNA. The DNA must be recovered from the cells or tissue of the body. Only small amount of tissue like bloods, hair roots, skin, etc. is required. Number second, restriction digestion. The isolation DNA is treated with restriction enzyme The restriction enzyme cut and DNA into small fragments having variable length. This phenomenon is called as restriction fragments length polymorphism. Number 3, gel electrophoresis. The DNA samples are loaded for agarose gel electrophoresis under the an electric influence. Number 4, Southern blotting. The separated DNA fragments are transferred to a nylon members or a nitrocellulose filter paper by placing it over the gel and soaking them with filter paper overnight. Number 5, selection of DNA probes. Known sequence of single strand DNA is prepared. It is called DNA probe. Number 6, hybridization. Probe DNA is added to the nitrocellulose filter paper containing host DNA. The single stranded DNA probes pair with the complementary base sequence of the host DNA strand. As a result, DNA hybrid are formed on the nitrocellulose filter paper. Remaining single strand DNA probe fragments are washed off. Number 7, photography. The nitrocellulose filter paper is photographed on an X-ray films by radiography. The film is analyzed to determine the presence of hybrid DNA and these DNA fingerprinting as shown in the figure. And last topic of this chapter, that is application of DNA fingerprinting. First application, in forensic science, DNA fingerprinting is used to solve problem of rape and some complicated murder cases. Second application, DNA fingerprinting is used to find out the biological father or mother or both of the child in case of disputed percentage. Third application, that is DNA fingerprinting is used in pedigree analysis in cats, dogs, horse and humans. Thanks to all.